the Helmholtz Institute for RNA-based Infection Research in the Barquist Lab for Integrative Informatics in Infection Research. With a background in elementary particle physics, she now works as a computational biologist on modeling functional genomics data with Bayesian statistics and simulation-based inference.
Yeah, thank you very much for the introduction and for giving me the opportunity to present my research here today. So I will talk about amortized Bayesian inference of bacterial cell cycle states and single-cell RNA-seq data with neural posterior estimation. So let me first tell you a bit about what kind of data we're talking about when we look at single-cell RNA-seq data. So, or not only RNA-seq data, but in general functional genomics data. So the kind of data can be genomic data. When the genome is transcribed, we get RNA, so we can be measuring RNA, or that's being translated to proteins, so we can also be measuring proteins. All in all, with functional genomics, we study the contribution of genes to cellular processes. And this provides insights into biological systems and disease development. The experimental setups can be very complex and have a lot of confounding factors, and that makes it a very good playground for Bayesian modeling. So more specifically for bacterial single-cell RNA sequencing, why are we interested in this? So when we have bulk data, we have in the bacterial population, we basically measure this as a uniform population. But this is not uniform. There can be some bacteria which have a stochastic gene expression or other gene regulatory networks switched on. And this may, for example, lead to antibiotic tolerant bacteria. Another important aspect is the cell cycle. So all these bacteria are different according to their cell cycle state. And my goal is to model the cell cycle effect. When modeling it, I can hopefully disentangle it from stochastic gene expression and thereby actually identify the cells which have stochastic gene expression and genes which lead to antibiotic tolerance, for example. And another goal is when I model the cell cycle, I can actually compare between different growth conditions and separately account for these cell cycle effects in both data sets. So it's important also for the integration of data sets. So what do I mean when I say cell cycle effects? So what's been shown is that during exponential growth in rich media, the doubling time is around 20 minutes, while the DNA replication time is 40 minutes. So one DNA replication cycle has to start before the first DNA replication cycle is finished. This means that genes which are close to the origin of replication will have a higher number, will be overrepresented in the cell. And since this is something that basically progresses throughout the cell cycle, it actually leads to a periodic gene expression pattern. So when we look at single-cell data of exponentially growing bacteria, we can then order the genes by the distance from the origin of replication, and we get these kind of pictures of the correlation of z-scores. So we can see this periodic pattern in there. And this cell cycle effect has already been described, but with a very complicated and heuristic workflow. And my goal is to develop a statistical model for it, which is reproducible and also applicable to other data sets, and which can also be integrated into analysis workflows to subtract the cell cycle effect. So the data set that I'm looking at are 30,000 E. coli cell. E. coli is an important model organism in microbiology. And it's exponential growth in rich media. So as I said before, doubling time is 20 minutes and DNA replication time roughly 40 minutes. In the data set, I also looked basically at you can see the correlation of the z-scores on the left. So that's the kind of periodic pattern which I want to describe with my model. The base distribution that I'm using for it is the negative binomial distribution because it describes well the overdispersion of the count data in RNA-seq data. And the important parameters are the gene-wise mean and the dispersion, gene-wise dispersion in this case, which can also be a gene-wise and condition-wise dispersion, so that can be made more complicated. And then to add the cell cycle effects to this, I introduce a form factor, which reflects this periodic pattern. So as the form factor, I want something that has a fast increase when the gene is being replicated, and then it has a slow decrease while other genes are being replicated. So I use, I tried this as a first try, and it actually immediately worked quite well. So I stuck to it for now. So this function depends on the gene position and on the cell angle. Well, by cell angle, I basically mean the cell cycle state where between two division processes the cell is. Then there are some global parameters, which are the strength of the effect or the factor between DNA replication and cell division time. And since I have 30,000 cells, this doesn't work with sampling-based approaches, so I'm developing an amortized simulation-based inference, where I simulate from a mechanistic model, then I train the neural density estimator on the simulated data, and I combine this with observations to get the posterior, and as a result, I get basically these figures on the bottom, where I get the gene expression pattern throughout the cell cycle. I tested that I can recover the cell cycle state in simulated data. So the data is quite noisy, which means I need a lot of simulations. And it also has its invariant under a 2π shift, so I get some hits in the off-diagonal corners. So that basically, as I said, the data is quite noisy, so I need at least 50,000 to 100,000 simulations to extract this one parameter. And also to show that it's really because of the noise and not because of a misspecification of the model, I simulated data with lower noise levels, and I could show that the root median squared error actually goes down. So I compared this to a fully hierarchical Bayesian model. Here, the global parameters are actually model parameters and are also fit in the model. I can apply this only to a subset of 2,000 cells, which takes about 3 days to run on a high-performance computing cluster. And yeah, I fit the global parameters within the model and use MCMC and the probabilistic programming language STAN. And as you can see, I get a good correlation between the simulation-based inference model and the fully Bayesian model. So to summarize, the results that I get are these gene expression is the gene expression throughout the cell cycle for different genes. And I showed also that I can get the cell cycle effect back from simulated cell angles. The Bayesian model takes about 3 days for 2,000 cells, and the amortized inference takes about less than 1 hour for 30,000 bacteria. That includes training and inference. So currently it's a fairly simple model that runs very fast. And the result are these is the gene expression across the cell cycle. And this is still a fairly young project that I started only at the beginning of the year. So now the question is, where do I go from here with this? So one thing is, for example, that I get these gene expression across the cell cycle, and some of them don't follow exactly this predicted cell cycle pattern. So it would be interesting to add more parameters to the model, also gene-wise parameters, which can parameterize this shift. So a non-zero shift could, for example, mean that a gene is involved in the cell cycle. And in order to fit this, or to make this computationally feasible, I'm, for example, thinking of factorizing the posterior into the part that comes from the cell cycle, or that basically fits the cell cycle model that I've already shown, and a second part which parameterizes the gene-wise shift or other gene-wise parameters. Another thing where which I can use this for now is if I want to subtract the cell cycle, or if I want to compare between different conditions. For example, if I have samples which are taking enriched and minimal media, they have a different factor between DNA replication and the cell division time. Or, for example, if I look at antibiotics and no antibiotics, because also in this case, without antibiotics, I would have exponential growth, and with antibiotics, I would probably see other DNA replication patterns. Then I can also investigate the effect of antibiotics on the cell cycle. For example, if I treat with antibiotics and DNA replication is stalled, I would expect all the cells from this data to accumulate basically at a certain cell cycle state. I can also try and see whether this has an effect on bulk RNA sequencing data. So try and identify these kind of cell cycle effects in bulk RNA sequencing data where single cells are being averaged out. And with this, I want to thank the Barquist lab where I work as a postdoc, and also former lab members, and you for listening to my talk.
Thank you for the presentation. Any questions from the audience? Peter.
Hi, Laura. I'm not sure. So you use SPI, and I really love that you showed that it's much faster than MCMC. This is how I understood you. Did you look into coverage tests of your posteriors? So you showed the PPCs, but maybe I just missed it. How well the posterior actually covers with respect to your labels?
I mean, basically, I did this on the one hand with simulated data. So I showed that I can retrieve again the cell angle in simulated data. But since I can also not be sure that the model actually accurately describes the data, there are basically two things which I did. So on the one hand, I actually verified that the distribution is fairly basically the distribution of the cell cycle state is fairly uniformly distributed. And I also tested other periodic functions, and when I take a simple sine function, for example, it's not uniformly distributed at all. And another thing which I think is a very good test for that it's actually doing something reasonable and extracting biology is that I really get these gene expression patterns across the cell cycle. Because this is something that's always for every gene that's again shifted by a gene-wise parameter. So if this was basically just some artifact of the modeling or something like this, I wouldn't be seeing this exact shift for every gene basically.
Okay.
So yeah.
Okay, thanks.
Well, maybe I have a final question. We have seen this triangle of compute, data, and methods now several times in this conference. What are you bound by? I mean, waiting for 3 days on compute, okay, that's probably not a problem. Can you comment on that? What is, if you scale this further up, if you generalize this further, what do you think is the limitation?
You mean what's the limitation in dataset side that I can apply this to, or what's the limitation in applications? I mean, or I can also comment on both. I mean, our goal is to really make this accessible and make this a method which can be integrated into an analysis workflow. And if we give this to someone in the lab and he has to wait 3 days for a result or even longer, that's too long. So it would basically, if we can wrap this up in a package which gives you a result within half an hour, then it probably has a chance to become part of an analysis workflow. Okay. Yeah.
Okay, thank you.